Hello, good day everyone. You're welcome to my YouTube channel, Pharmaco Healthy channel. My name is Pharmacist Goodluck, and in today's presentation, we are going to address a common misconception that does not only exist among the general public, but also exists among a handful of healthcare providers. What is this misconception that we are talking about? We are talking about the misconception that diclofenac potassium is better tolerated than diclofenac sodium in ulcer patients. This statement is inaccurate, and during the course of this presentation, you are going to identify by yourself why diclofenac potassium is not in any way safer than diclofenac sodium when managing pain in ulcer patients. So, having said this, we are going to delve into the presentation proper. But before we do that, I suggest we go for a short break. So, if this sounds so appealing to you, I advise you to stay tuned. Don't go anywhere because I'll be right back soon. Hello everyone, you're welcome back to my YouTube channel. So the question now is, what is even diclofenac? Before we now talk about diclofenac sodium and diclofenac potassium. So chemically speaking, diclofenac is a monocarboxylic acid consisting of phenyl acetic acid with a 2,6-dichlorophenyl amino group at position number 2. Basically, diclofenac is used as an analgesic agent for management of pain, number 2 as an antipyretic agent for management of fever, and then number 3 as an anti-inflammatory agent for management of inflammatory conditions in the body. It has been found out that most drugs in their free uncomplexed form are usually weak bases and weak acids with poor pharmacokinetic profile. When we say pharmacokinetic profile, we refer also to the physicochemical properties of the drug. So diclofenac in their free uncomplexed state are usually monoprotic acid with poor solubility, which in turn affect drug dissolution rate, bioavailability, and drug onset of action. So because of this, these drugs are usually formulated in their salt form called pharmaceutical salts, and that is the origin for diclofenac sodium and diclofenac potassium. It has been found that in addition that even though these drugs are formulated in their pharmaceutical salt, their physicochemical properties can still be further manipulated by simply interchanging the counter ion that has been used in formulation of these salts. This characteristics has been employed by pharmaceutical company to generate different formulations of drug with the same active pharmaceutical ingredient but differ in their pharmacokinetic profile for management of a plethora of medical conditions. From our basic knowledge of chemistry, a salt is a product that is obtained when an acid reacts with a base in a type of reaction called neutralization reaction. In pharmacy, a pharmaceutical salt is an ionizable drug that has been combined with the suitable counter ion to form a neutral complex. This neutral complex is now the same salt that we spoke about in our basic knowledge of chemistry. This concept of pharmaceutical salt is one of the applications of our basic knowledge of chemistry in drug manufacturing processes to solve real-life problems. The suitability of a particular counter ion for utilization in formulation of pharmaceutical salt generally depends on the physicochemical property of the active pharmaceutical ingredient of the particular drug that we are putting into considerations. One of the common criteria that has been utilized by drug manufacturing companies in choosing a particular counter ion for formation of pharmaceutical salt is to use those counter ion that has been used in a drug that has been FDA approved. So this is now regarded to be generally safe for utilization. The prediction of the properties of the pharmaceutical salt for both the qualitative and the quantitative property based on the counter ion used is an important research area. So I will advise our young researchers to invest their time, their energy in this area of research, because over the years it has been a hard nut to crack by our researchers. So who knows, you might be the one to break this barrier and be able to predict with some degree of certainty and clarity the quantitative and qualitative properties of salt formed based on the counter-ion that has been utilized in formulation of this pharmaceutical salt. The most commonly used counter-ion for basic drug entity include hydrogen chloride, hydrogen bromide, acetate, fumarate, and mesylate. However, the most commonly used counter-ion for formulating drugs that belongs to acidic drug entities include potassium, sodium, and calcium. Now, having said this, we are now going to delve deeper into explaining the reasons why these drugs are formulated as pharmaceutical salt. Even though one of the most monumental reasons why drugs are formulated as pharmaceutical salt is to address the issue of poor aqueous solubility, however, these possibilities can also be utilized to address other physicochemical and biological concerns associated with the drug. Hence, drugs are formulated in their pharmaceutical form for the following reasons. 1, to reduce the toxicity of the drug. Number 2, to improve chemical stability of the drug. Number 3, for easier drug administration. Number 4, to address the concerns associated with drug manufacturing processes. Then finally, just like we have said before, to give room for manipulation of the pharmacokinetics profile of a drug, like the solubility and dissolution rate, which in turn is going to affect drug bioavailability and also onset of drug action after it has been administered orally. Having said this, I would like to make this concept clearer by giving examples of drugs formulated in their pharmaceutical salt and the reasons why they have been formulated in their pharmaceutical salt. So number 1 is propoxyphene. Propoxyphene is a drug that belongs to the class of drug called analgesic. It was initially used in management of pain. Even though this drug is currently withdrawn from the market due to its ability to put patients on high risk of cardiac arrhythmia, permit me to use this drug as an example to explain the concept of pharmaceutical salt, because it is a good candidate that will enable us understand this important concept. Okay, so propoxyphene was formerly formulated as propoxyphene hydrochloride, and propoxyphene is usually administered with aspirin. So it was observed that when propoxyphene come in contact with aspirin, it starts undergoing degradation. So because of this, propoxyphene was now formulated as propoxyphene napsylate. And guess what? After the formulation as propoxyphene napsylate, it was observed that this degradation associated with propoxyphene when it comes in contact with aspirin disappeared. Okay. Additionally, it was also found out that the acute toxicity of this propoxyphene was reduced by half. That is the beauty of what we are talking about. So number 2 drug that we're going to talk about here now is perindopril. Perindopril is an antihypertensive drug that belongs to the subclass angiotensin converting enzyme inhibitors. Perindopril is used in management of hypertension and other related cardiovascular conditions. It was found out that perindopril initially was formulated as perindopril albumin. So this perindopril albumin is highly unstable, and before it could be exported to countries with high relative humidity and high temperature, it requires a special packaging. So because of this, perindopril has now been formulated as perindopril arginate. And when perindopril arginate was compared with perindopril albumin, the stability of perindopril arginate increased, or rather improved, and also the shelf life of perindopril arginate also increased by 50%, thereby making it possible for perindopril to be exported to countries with high relative humidity and temperature. So number 3 is diclofenac. Diclofenac, as we have established before, in their free uncomplexed form, have poor physicochemical properties, or rather poor pharmacokinetic properties. So even in their pharmaceutical form, they still have variable pharmacokinetic properties. As a matter of fact, the significant difference that exists between diclofenac potassium and diclofenac sodium is that the former has better aqueous solubility, which in turn improved the drug dissolution rate, bioavailability, and onset of action. That is to say that diclofenac potassium have a better onset of action when compared to diclofenac sodium. The research carried out to compare the onset of action between diclofenac sodium and diclofenac potassium, actually it was discovered that it took diclofenac potassium less than 1 hour, about 30 to 54 minutes, to attain peak plasma diclofenac concentration when compared to diclofenac sodium that it took about 2 to 4 hours to attain this mean plasma peak diclofenac concentration. So this fast onset of action could be used to explain the reason why diclofenac as powders for reconstitution or tablets for dissolution, all targeted for oral injection, are formulated as diclofenac potassium and not diclofenac sodium. Example is Voltfast. This could also explain the reason why diclofenac in their formulation as sustained release drug are formulated as diclofenac sodium. This is seen in clofenac SR 100 mg. This could also be the reason why we, the healthcare providers, also advise that patients that are taking diclofenac in normal 2 to 3 times daily dose to relieve long-term pain should use diclofenac sodium rather than diclofenac potassium, because the fast onset of action of diclofenac potassium may render this diclofenac clinically irrelevant in this kind of patients. However, for patients that have the ambition to achieve fast relief from acute pain, should use diclofenac potassium, because diclofenac potassium has a faster onset of action and will give immediate effect. So finally, I would like to say that patients that have ulcer should avoid diclofenac potassium as much as they avoid diclofenac sodium and other NSAID. This is because diclofenac potassium only differ from diclofenac sodium in their salt content, hence their pharmacokinetic profile, particularly their solubility that has affected the drug dissolution rate and the drug bioavailability, and also the onset of action, but not in their mechanism of action. Generally, diclofenac potassium and diclofenac sodium belong to the class of drug NSAID, and NSAID act through inhibition of prostaglandin synthesis. This inhibition of prostaglandin synthesis is the reason why NSAID precipitate ulcer. So both diclofenac sodium and potassium are definitely going to inhibit prostaglandins, thereby precipitating ulcer. Having said this, I would like to ask, did this video add any value to your knowledge? If yes, go to my YouTube channel, click on the subscription button. But if no, I am sorry. I will definitely make it up to you in the next post I am going to make. But one thing you still need to do for me is to go to my YouTube channel, click on the subscription button, put your notification button on. Also, don't forget to follow me on all my social media handle. Why are you doing all of these things? So that the next time when I post this video I'm preparing for you, you will not miss out. So at this point, I would like to say thank you for your audience and God bless you. See you in my next episode.